Okay, good afternoon. My name is Dele Ogunsheton, and I welcome you to our public health seminar series. This is a continuation of quarter-long, actually year-long, and we typically have them every other Mondays, but this quarter it's been every Monday because we've just been blessed with distinguished speakers for the series. And we are delighted to welcome those who are joining us for the first time, and for the regulars. We appreciate your continued support. This series is possible because many of our faculty members build a network of colleagues all over the country and the world. And it takes us a while to get some acclaimed speakers. As for today's speaker, I think it's been a couple of years since we've been trying to get her to come to UC Irvine. And thanks to the persistence of our faculty members who nominate speakers. And today's speaker was nominated by Professor Tim Allen Bruckner. He's assistant professor in public health and in planning, policy, and design. And he works in all aspects of epidemiology, health policy, and teaches courses in health policy and management and community health. Many of our students who have not met him, I welcome you to visit his website. If you're interested in his work, please send him an email. He's very busy, but I'm sure if your interests coincide with his, he would be more than delighted to talk with you. After the presentation, we invite everybody to lunch with the speaker. I say that with some trepidation because I know not everybody will show up, but one day everybody will show up and I'll be in trouble. But the lunch is a way, it's a way to continue the discussion with the speaker. And we hold that at the lobby of the Anteater Instruction and Research Building, just about 5 minutes walk from here. So I give that long introduction to make sure that everybody who's kind of walking in get a chance to settle down. This hall has very good acoustics, which means the sound carries, and if you're tapping on your computer or have to leave your seat, please do so as quietly as possible. So now please welcome Dr. Tim Bruckner to introduce our speaker for today.
Hello. All that energy for Irva. So it's my distinct pleasure to introduce Hertz-Picciotto to you. I just learned this morning, actually, that she started her undergraduate studies at UC Irvine. So she is an anteater at heart. Dr. Hertz-Picciotto is a professor of epidemiology at UC Davis, and she's also the chief of the Division of Environmental and Occupational Health. She received her M.P.H. and Ph.D. at UC Berkeley. After her degrees, she started a faculty position at UNC and was there for 11 years and then went to UC Davis. She has over 200 publications, a very productive and impactful scholar, and today she'll be talking about autism. I want to point out a few other things about Irva. In 2011, she was awarded the John Goldsmith Lifetime Achievement Award for Outstanding Environmental Epidemiology by the International Society of Environmental Epidemiologists, which essentially recognizes her strong contribution to the field. But as a personal side, in addition to these accomplishments, she's also a very friendly colleague and mentor. So please join me in welcoming Dr. Hertz-Picciotto.
Should I bring it close? Wow. Okay. I hope that didn't blast your ears. Thank you very much for the introduction. So I've been at UC Davis now for 10 years, and during this time I have just taken on this whole new area of research on exposures from the environment and their relationship to autism, which I'm going to talk to you about today. Before that, I used to study environmental exposures and their effects on pregnancy and pregnancy outcome, but this was a really great opportunity when I came to Davis because of the MIND Institute, which stands for Medical Investigations of Neurodevelopmental Disorders, and it was a new institute just getting started as I was making the move. In fact, it's part of the reason I made the move. So what is autism? In 1943, Leo Kanner, who was a psychiatrist at Johns Hopkins, published a paper in which he described 11 cases that had come to him for psychiatric evaluation, and he documented these patterns that are in 3 domains, which in our current, well, I should say our most recent, but no longer current, DSM, Diagnostic and Statistical Manual for Mental Health Conditions, remain the definition of autism. These defining features are a lack of social reciprocity, a deficit in communication skills, and a pattern of repetitive behaviors or restricted interests, what Kanner called an insistence on sameness. And the definition also says that the symptoms are generally present by 36 months of age. It's considered to be lifelong without a cure. But this is actually quite controversial, and there are documented cases of people who started out seeming to meet the definition who seem to actually outgrow the diagnosis. So we have this umbrella of pervasive developmental disorders that includes all 5 of the conditions shown on this slide. And then within that, these 3, autistic disorder, Asperger's syndrome, and pervasive developmental disorder not otherwise specified, is what's usually referred to as autism spectrum disorder. And in the new DSM-5 that, as I understand it, is now effective, there are actually no distinctions being made amongst these 3. So that may actually end up having some impact on the counts that we start hearing published about what the incidence and prevalence are, because some studies have taken a more strict definition than others, but this will now be the standard. The diagnosis is made, there are many instruments, but these I think are considered the really gold standard, reliable instruments where reliability has been established, and that's really been a major contribution coming from the clinicians in terms of developing these standardized instruments. So the first one is the Autism Diagnostic Observation Scale, and this is about a 20 or 30 minute interaction in which the evaluator has these presses, and they're scripted, and typically developing children will respond in certain ways related to communication, their imaginative use of play materials, and I'll show a little clip of an ADOS being administered. And then the ADIR, the inventory, is an interview with the primary caregiver, and it's historical. It's giving kind of the trajectory of the child, through early years of development. And it's lengthy. It takes 2 to 3 hours to administer that. Both of these provide scores on the 3 domains that I talked about. So let's see. This is a clip of Leslie here is administering the ADOS to the child. So she's got this little Play-Doh birthday cake. And she was saying, she was lighting the candles. And this is the little baby here, the doll, whose birthday she's pretending it is. And you can see the child is totally ignoring this whole situation, not engaging. A birthday party for a kid this age would be an exciting thing. Even, pretending it would be an exciting thing. And now she's saying, okay, we need to feed the baby a little piece of cake. And he's not really playing with the idea that this is a cake. He's just sticking to the idea it's Play-Doh and I'm going to stick in. So then he finally puts the piece of it on the spoon. He actually drops it, goes down, picks it up. And now he engages in this idea that maybe it is something you can eat, but He wants to give it to himself instead of to the doll, right? So that's, okay. So this is, sort of lack of social engagement. You notice he never looked at her face, he never looked at her eyes, he never really interacted with her either directly or indirectly through the play activity. So facts about autism: it's got a 4-to-1 male to female ratio. The prevalence in the U.S., the latest statistics out of the CDC is 1 in 88, which is 1 in 58 for males. There is a really sordid history, I would say, about the beliefs of what were the causes for autism, and since the 1960s to about the 1980s, the paradigm that was really accepted in the professional community was that this was bad parenting. This was due to cold parents who, refrigerator moms was one of the expressions that was used, that their parenting style was not bonding with the child, and that's why the child had this problem. And we know that that is absolutely not the case, yet I think there is always a legacy that sort of lasts beyond long after that has been really proven not to be true because of the postmortem autopsy studies where they've really seen there are lesions in the brain. There are electrophysiology studies and, of course, MRI and functional MRI studies that really show the brain the connectivity is very different. But there continues to be, I think, some aspect in which the clinical community and the professional and the medical community has not fully engaged parents in some of the research kinds of questions of the day. And some of the ideas that may be very popular in the community have been, I think, dismissed with not so much the scientific way, but disdainful of parents for having those beliefs. And I think that continues some of the friction, I think, that there is between the communities and the scientific world. So yes, we know there's a neurologic basis to this disease. Genes clearly play quite a sizable role, but so do environmental factors. And that statement itself is controversial, or has been. For many years, and in fact I still hear my colleagues saying it, the early genetic studies seem to suggest a very high correlation between monozygotic twins and a very low one between dizygotic twins. So this was used to calculate heritability, and the estimates in some of the very early studies were 70 to 90%. There's actually one where I guess it was 100%, and I recently heard that figure being quoted. In fact, there's a new issue of Nature that has a whole section, it's called their sponsored section, where all of these number of foundations and organizations that have been involved in autism research contributed articles, and those articles basically quote this one 1985 study that says it's 100% heritable. Since then, there have been some very large studies that have brought that number down, and the two largest studies seem to give numbers of around 38 to 58% heritability, which suggests that genes and environment both play really important roles in the causation of autism. And I think that's important because the idea of intervening to really prevent autism requires that there be things that are modifiable. Genetics, it's a little bit harder to modify, so it gives, I think, a public health message that there may be things people can do, and we'll talk about those. I also want to emphasize multifactorial causation, which is this idea it isn't genes or environment, but in fact the two together that combine to really mold what happens in terms of really all health conditions. So why not autism? And multifactorial means that not only across the population different causes, but in fact even within each individual there are probably multiple genetic and multiple environmental factors that come into play, and so that's what these little pies of causation represent, that you take any one of those factors away and you no longer have a sufficient set of causes to bring about that behavioral syndrome, that development. And then I also want to just add to the multifactorial idea is that there's a developmental period where the brain is particularly susceptible, and some of the really important time points may be very, very early, around the time of gametogenesis and conception, and shortly thereafter. This appears to be a very intensively vulnerable period, and there's different lines of evidence supporting that, including an association with paternal de novo mutations that would in fact be the result of exposures of the father in that preconception time period. And then there are a number of studies suggesting that during gestation, particularly the beginning of gestation, but not necessarily excluding events that happen around the time of birth as well. So, and postnatally, in fact. I don't think we, at this point, can preclude some role of postnatal factors. So things can go right or they can go wrong. This is just a list of potential causes for autism, and some of these I'll be talking about data that support, and some of them data that don't support, and some of them I won't be talking very much about. So I'm not going to say a whole lot more about genes, except that we know there are many, many genes that are involved, and the landscape has really changed. I think 10 years ago people were saying we're going to find the gene, and now people are saying there's going to be hundreds of genes that may be involved here. Environmental chemicals, that's an area of great interest to me and my research team. And I'll talk a little bit about metals. Pesticides, there have been several studies now suggesting associations between pesticide exposures and autism. One study seems to suggest it's organophosphates, another one suggests organochlorines. I think a lot more work needs to be done, but there are clues. And the thing about pesticides you've got to understand is, what are pesticides about? We design them to be lethal to, certain species, and many of them work by attacking the central nervous system. Vitamins I'll be talking about, and they're the original finding, possibly the first environmental factor identified for autism, was the congenital rubella. There had been an epidemic in the 1970s, and there were 2 different independent studies suggesting that those congenitally exposed children had a substantially higher risk for autistic features. And then complications during pregnancy, that I'll also say a few things about. Fertility treatments and other medical interventions have been proposed. The studies on the fertility treatments have not been all that convincing. Again, though some of the numbers have been very small when you get down to specific ovulation-inducing drugs or in vitro fertilization and so forth, separating them out. The other medical intervention that's been proposed is ultrasound. We certainly have had a rapid increase in the use of ultrasound, and earlier and earlier in gestation. And ultrasound delivers not an X-ray, but it's a thermal index, a thermal dose, and there's a lot of biological evidence that high temperatures that can have impact on brain development. So, and then other medications have been of interest. SSRIs, there have been some studies suggesting that SSRIs during pregnancy may increase risk. Maternal reproductive characteristics and everything else, cell phones, you name it. So lots of hypotheses and not nearly enough data. Also want to mention the time trends. So between 1990 and 2001, autism incidence actually rose by about 7-fold, so a 600% increase in risk. And we published an analysis of that for the California data which showed that about 200% of that 600% could be explained through various artifacts like the change in criteria that happened back in the early 1990s, where it went from DSM-3 to DSM-4, the trend towards younger age, broadening to include milder cases, and then delayed childbearing. So it doesn't explain the full 600%. It does explain some of it, and other people have looked at what we call diagnostic substitution, where a child 10 years ago or 20 years ago, we might have said, oh, this is a kid who has mental retardation. Now we look at that child, that same child, and we say, oh, they've got autism. So certainly, it doesn't explain the full 600%, which I think adds to the reasons to be looking at environmental causes, because some of this increase may in fact be real. And as a thought experiment, I'd encourage thinking broadly. If autism is going up, what other things are going up: asthma, obesity, diabetes, ADHD. And should we be thinking broadly about what kinds of biological changes, environmental changes have happened that affect biological systems that could actually be related to more than just autism? So about 10 years ago, I started working with a group at UC Davis, and they said, well, we'd like to have an epidemiologist join our team, and would you like to join us in applying to establish a children's center focused on autism? And I said that I didn't know anything about autism, and they didn't really blink an eye, and they just said, that's okay, nobody does. So we started out with the CHARGE study, the Childhood Autism Risk from Genetics and the Environment, and set the goal of trying to identify causes and contributing factors, and looking at environment and also gene-environment interaction. And we thought it might be useful as well to think about autism as maybe having different subtypes that themselves may be etiologically different. So we work on a lot of different levels, biologically and behaviorally. And we put together kind of a list of where do we begin, since there's really virtually nothing known at that point in time except this congenital rubella, and then also some connection to thalidomide. And this is kind of the list we came up with, and ways to collect information about these factors are listed on the right. And so, if you're interested in metals, you can collect blood. You can also look at hair. You can get information about the diet. If you're interested in viruses, you can look at medical records, and so forth. So this was we designed it as a case control study. To do a cohort study would have been really outside the range of what one could do with the budget we had. With 1 in 88, that would be 87 non-cases for every one case. And we focused on trying to recruit from the population, which was facilitated by the fact that California has this Department of Developmental Services, which people go to get services for people who have developmental disabilities. And they have collaborated with us in making available the list of persons who are eligible for services in California. We also do standardized confirmation using both the ADOS and the ADIR, and I'm working with a group of molecular biologists, immunologists, people with a range of laboratory skills, so that it could have a molecular component as well. Our cases are children with autism or children with developmental delay. That's another case group, or it could be looked at as a control. And then we have the typically developing controls. We select using birth certificate files children from the general population, and we frequency match to that 4:1 male to female ratio, and also age and broad geography. And then we bring them all into the clinic to evaluate and make sure they're diagnosed in the category that they really belong in. And some of the children who come in as developmental delay, it turns out they actually really do have autism, for example. So the three groups, all of them have to be 24 to 60 months of age, born in California, living with a biological parent who speaks English or Spanish, and within our catchment area, which is, the Mind Institute is in Sacramento, and then we have all of these green counties, the pink counties, this yellow county. Basically, it's an hour-and-a-half drive to the Mind Institute. And we assess the child not just for autism, but for other developmental, cognitive, adaptive skills, deviant behaviors, and so forth. Then we conduct an interview, a lengthy interview about the environment and the child's preconception and gestational exposures. And we have this index period 3 months before conception all the way through the first year of life. We collect specimens from the child and the family members, and then we get the medical charts as well. So, and this is our slogan to the families: be in charge. So, starting with results now. Metals we looked at, we wanted to see whether current blood mercury levels differed in children with autism versus typically developing children. And we found that the typicals actually had slightly higher levels than the children with autism. You can see the mean there and how it's shifted to the right a little bit in the top panel. So the lower levels in children with autism and developmental delay, as it turns out, was because they ate less fish, and mercury is, of course, most of what we intake, most of the intake from mercury is actually from fish, methylmercury. So if they were eating less fish, they had lower levels. And when we adjusted for their fish consumption, we actually saw no difference between cases and controls. That's exemplified by the top p-value up there and the 4% difference between the two groups. Interesting, the developmental delay group actually still had, even after adjusting for the fish consumption, still actually had lower levels of mercury, and I'm not quite sure what that's about. But the fish consumption clearly is related to higher levels, you can see that. Also, mercury amalgams in children who either chew gum or had a like grinding habit, and not if they had the mercury amalgams but didn't have the grinding. They had to have both of those to actually have higher levels of mercury circulating in their blood. All right, so the children with ASD did not have higher blood mercury, and in fact there was no difference after we adjusted. But this really doesn't address a causal question of whether mercury causes autism, and that has been one of the questions in the community. And you might say, well, why did we look at it? Well, the reason we looked at it was because some people were suggesting that the differences were because these children didn't metabolize mercury properly, and that it was really sequestered in the brain and so forth. They were really holding on to it. And in fact, it seemed not to be true at all in our data. The levels were quite comparable to national data from N. Haynes' survey. So it doesn't seem to be a metabolism issue. And we've done some more work looking at that. And it really, in terms of the way the fish consumption is related to the blood levels, is really consistent with nothing unusual going on here. We have done some analysis now of baby hair that the mothers were asked to bring in, which is usually the first haircut. A lot of people save the first haircut from their baby, and so about a third of the women brought those in, and we've been measuring mercury in that. And we also have newborn blood spots that the state of California archives and makes available for researchers. So we are still planning to look at the causal question as well. All right, air pollution. We were interested in whether air pollutants, which includes compounds like polycyclic aromatic hydrocarbons, metals, and so forth, might be at higher risk for having a child with autism. And we looked at initially the proximity of their homes to freeways, which is actually a very good indicator of traffic-related air pollution. So this slide shows that the top group, which were the closest 10% of the population in terms of their distance to the freeway, actually based on the control distribution, were at almost twice the risk of having a child with autism, and that there wasn't any rise as people got closer. If you start with the referent group being people who are almost a mile away, and so there's not a dose response, it's just this group. And as it turns out, the data on air pollution suggests that once you get the distance that this group is, which is about 300 meters, the levels of traffic-related air pollution actually drop down to background levels. So, in fact, it's consistent with what we know about dispersion around freeways. The nice thing about this analysis is it is very much an objective measure of exposure. It's not relying on people remembering what they did. And that this metric has also been associated with asthma. And then also the components of air pollution have been shown to induce neurodevelopmental deficits in rodents. So these are important compounds. I would add, however, the data on cigarette smoke has not been as consistent, and that's actually really, I think, a conundrum, because the same chemicals are in cigarette smoke that are in air pollution. So I think we really need to resolve whether there's problems with the cigarette smoking studies, or is there some problem with the air pollution studies, although there are now several other air pollution studies that seem to be showing these associations. And I will say I was very skeptical when we started looking at this, but it looks like it's showing up. We need more data, though. So turning now to nutrition, folic acid is required for synthesis of DNA and proteins, and we recommend that women take folic acid starting before they become pregnant to prevent neural tube defects. That's the reason. We also have fortification of our cereals and grains. And since that happened, we are seeing a decline in neural tube defects, which is, of course, a good thing. Well, we looked at prenatal vitamin supplementation in these women, and what we saw was a lower likelihood of developing autism in the mothers who took the prenatal supplements, of about 40% reduction, so an odds ratio here of 0.6. And so here are the cases were less likely and the controls were more likely in that very early period, the 3 months before pregnancy and the first month of pregnancy. No difference. By the time people realize they're pregnant, most of them are taking the supplements, but the real difference here is in that very early period. And of course, why is folic acid important? It is important because it is a methyl donor, and so this slide actually is here to point out a couple of genes that regulate the conversion from folic acid to folate, and then that are involved in this epigenetic kind of mechanism. And what we were interested in was whether your efficiency in this one-carbon metabolism cycle was related to, in combination with their prenatal supplementation, whether that influenced risk for autism. So gene-environment interaction. And what we found is that if you have the high-risk genotype for this particular gene and for several others in that were up on that slide, MTHFR being one of them, there was this combined effect. So if you didn't take the supplements and you had the more efficient transmethylation, you have some increase in risk, but if you are a poor transmethylator, you have a 7-fold increased risk relative to those who both took the supplements and the child had the favorable genotype. So this was one synergistic example, but several of the other genes also showed this. Which, by the way, is actually the first gene-environment interaction, I think, in this field that's been published. Okay, turning now to maternal metabolic conditions. We know that mom is really important to baby. Everything she takes in, roughly everything, most things, go right through the placenta. And one of my graduate students was interested in looking at maternal metabolic conditions like obesity and diabetes. And these are a couple of possible pathways that they might affect brain development in the child. So on the left side is a pathway that is a pro-inflammatory pathway where IL-6, for example, which crosses the placenta, actually results in a stimulation in the brain of the fetus, resulting in a pro-inflammatory response in the fetal brain. And then on the right side is sort of more focusing on glucose metabolism and increased fetal insulin, resulting in what might be a sort of fetal hypoxia, which also could result in neuronal damage. So two possible pathways that these conditions might influence the brain development. And when we looked at the data in the epidemiologic study, we did see that having one of diabetes, hypertension, or obesity, or one or more, I should say, resulted in about a 60% increase in the odds of developing autism, and even higher for developmental delay. And I didn't share the data on some of the other things I looked at, but we also saw that for the folic acid, that it influences developmental delay generally, not just autism. The hypertension, we didn't even have very many with hypertension, but you can see that seemed to be really strong. Obesity was the big driver in the sense that most of these women, it was obesity was the condition they actually had. Now, there have been some prior studies on diabetes and neurodevelopment. So this isn't a totally, from the cognitive development point of view, it's not new. But we also need to think a little bit about the metabolic conditions and the causes of them. So that's actually an area that we've actually applied for funding, and I think we're going to get it, to start looking at what kinds of upstream environmental factors influence maternal metabolism, which may be then the pathway for exogenous factors to influence brain development. And there are, in fact, some cross-sectional studies, but not necessarily cohort studies, that do indicate there are endocrine disruptors that increase or that are associated with diabetes and obesity, and other environmental factors like high fructose corn syrup seems to alter, is metabolized very differently than cane sugars. So things can go awry. And one of the interesting collaborations I've had is working with a group that has been looking at immune abnormalities in these children. And Dr. Judy Vandewater is an immunologist who has looked at these blood specimens that we've provided to her from the cases and the controls. And, when I first started working with her, I thought, people who find the result everywhere they look, you kind of get a little suspicious. Maybe their methods aren't very good. They're just, they get kind of twisting the data or something. But she is a really rigorous scientist. And there is a sort of widespread, subtle, sometimes not the thing you would see in a very small study, but in our sample sizes of hundreds of kids in each of the groups, we do see some patterns that suggest immune regulation is suboptimal in children with autism. And so we see lower overall IgG. There was also a little bit of a reduction of IgM. The subclass IgG4 was increased. TGF-beta is a regulatory cytokine, which was reduced, and so forth. So many, indications that something is not exactly the way it should be. And then we've been looking at plasma cytokines and looking even at the newborn blood spots, and again, seeing some differences. And that's on the child side. Now, this slide is actually looking at the mother, and it presents the data on maternal antibodies to fetal brain tissue. So these are Western blots, and there are certain patterns that we've started seeing in mothers of children with autism that were not seen at all in any of the mothers of children with typically developing children. So we're seeing this in about 6 to 12% of the case mothers, and it's this is the 73 kilodalton, and this is 37. They also found another one at 39 kilodaltons, different size, and they've now identified what these proteins are. We also I wasn't involved in this because I don't do primates, but Dr. Vanderwater and a couple of people at the Primate Center at Davis took purified IgG and injected it into pregnant rhesus monkeys and looked at the offspring. So this is really a controlled experiment. And the baby rhesus monkeys had these patterns of hyperactivity and lack of really identifying with their own mother. They were put in this experimental scenario where there's another mother from the play group that they bring these monkeys together, and they raise them and bring them together a few times a week in the play group. And the monkeys that had gotten just the saline, or in fact it was IgG from mothers. Sorry, it was not saline. It was IgG from human mothers who are typically developing. Those monkeys, they run around a little bit, and then they see their own mom, and they this is a post-weaning kind of experiment, and they come and they settle next to their mom. But these other monkeys that had been treated with the IgG from human moms who have a child with autism were just circling around, and then in another experiment they're in there with one of their playmates, and they start doing these motor repetitive backflips and all sorts of unusual kinds of behaviors, and the playmate is kind of looking at this other monkey, kind of like kids on a playground when a kid comes in who's a little bit weird, and children will, kind of back away and avoid them. So it was a very striking thing to see this video that you didn't need to calculate a p-value. It was so extreme, the differences, and suggesting that there is a neuroimmune connection going on, and that perhaps in some of these cases there's been some loss of the tolerance that pregnancy induces to allow the foreign body to grow. And so we're continuing to look at this as well. And of course, it's not clear. It could be that the alteration of the brain, is affecting the immunology, but the experiment in the monkeys really seems to belie that type of explanation. And then the last finding I'll show is related to maternal infection and fever, and this is again going back to the CHARGE study, where we had the mothers who reported they had fever and took something for it, mothers who reported they had fever but did not take any kind of anti-fever medication, any sort of antipyretic, and then those who did not have fever at all. And that was the referent group. And what you see is there was actually, it was limited to those who had fever and did not take anything for it, which seemed to be the group where you have an elevated risk for autism. And in fact, there's been another study now that came out of Denmark, I believe it is, that had looked at hospitalizations related to a prolonged fever. and also seem to see a connection to autism. And of course, that's a problem, could be a problem of self-report, although self-reporting the fever, but then why would the bias only be related to those who didn't take something for it? It's consistent with some seasonality data suggesting that maybe influenza or the inflammatory response that you have when you get the flu, and the winter months have been the months of higher rates of autism. So the significance, I think, is in this really first comprehensive study of environmental factors in autism is that earlier studies had used really kind of ecologic designs, most of them. So this one was really population-based, and I mentioned these other attributes of the confirmation of diagnoses. And then we are focusing on modifiable risk factors, which is actually not what most of the field is focusing on. There's really a huge effort to try to map the genetics of autism with really huge amounts of NIH money going for that. But the limitation is that sometimes we have to rely on self-report of the mothers, so that does have its limitations. And for that reason, we've launched now a prospective study of high-risk younger siblings. So it's a mom who has a child and is now getting pregnant with another child, and we follow the pregnancies in great detail, lots of specimens, lots of interviews, until the child's 36 months of age. So Marbles is one of several other similar, well, not necessarily similar, but prospective studies that complement the case control design and eliminate the problem of differential reporting, and also, of course, allow you to collect in real time specimens and information. These are some of the things we're looking at, analyzing now, or have a paper that's been rejected a few times and is still trying to find the right journal. Things about epigenetics, copy number variation, interactions, pesticides, medications. And then we have a paper that's actually in press now. It's our first paper from Marbles looking at some placental markers that I think will be interesting. And that's really about it. These are my collaborators and investigators from many places. And we have a huge staff working on these studies. And then, of course, none of this is possible without the generosity of the families who come into the clinic and spend a lot of time answering questions they find boring sometimes. And then we have funders to thank as well. And you. So I guess we have some time for questions.
I was wondering whether, thank you first of all for that wonderful talk. I was wondering if any of the environmental factors that you've examined and that seem to be associated with increased risk of autism point to why there might be this great sex difference with boys much more at risk than girls.
Yeah, that's kind of a question that people talk about a lot, and there's a lot of theories around that. A lot of heat and not a lot of light, I would say. Simon Baron Cohen is who's in England, has a theory that autism is the extreme male brain, and that he has these scales, the empathizing and the systematizing, and that the males, in general, males tend to be more on the systematizing and females tend to be more on the empathizing side in society at large. And that what's happened in these children is they've kind of lost whatever empathizing men may whatever level they have, they lack even that, and then they're more extreme on the systematizing side. Although he doesn't exactly, he's had a couple of papers suggesting that this has to do with hormone imbalances during the gestational period. And there was a study that one of his doctoral students did where and they weren't looking at autism, but they were looking at these scales, and they took amniotic fluid and they measured fetal testosterone, and seemed to see, higher fetal testosterone levels associated with more of that systematizing. So, there are some theories. When I showed that paper to an endocrinologist, he said, well, this is ridiculous. The time point that they took the fetal testosterone shouldn't really have any influence, and he didn't really believe it. And he's really a world-renowned endocrinologist, so I take what he says seriously. But it does, maybe that correlates with what happened at the right period in time. That's one of the theories. And then, that also leads to the possibility that maybe endocrine disrupting chemicals could play a role that have altered. Yeah, hi.
So I just want to highlight something that Irva is participating in. We formed a special interest group to look at whether or not abdominal ultrasounds on pregnant women increase the risk for neurodevelopmental problems in the offspring. And the literature suggests that there may be some type of association because, as Irva said, the ultrasound generates heat. And if you've got a very susceptible fetal brain coming in contact with heat, that may be a multifactorial reason for the development of other ADHD and things like that. So I just wanted to bring this to the group. This was a thought that I had and got in contact just to give you some just clarity on this. Somebody gave me a point of contact of Dr. Brian King, a child psychiatrist at Seattle Children's, who's in charge of the Autism Center, and asked whether or not he thought this was a crazy idea or not. And he said, I just want you to know that at this point in time, I'm studying pregnant rats, exposing them to ultrasound, and see if the rat's behavior is affected by abdominal ultrasound during pregnancy. So the thought that we have is that maybe something that we're doing, that we're taking for granted, may be causing some problems. And there's 3D, 4D ultrasounds, all kinds of activities that are going on. And we're not saying there is an association, but asking the question whether there is or not. And Irva's participation has been wonderful.
And this is Eric Handler, for those of you who don't know, is the county health officer for Orange County.
With regard to some of the environmental factors that you're looking at, and especially in relation to inflammation, are you looking at dietary factors as well, especially with some of the emphasis and things that have been looked at with regard to gluten sensitivity, wheat sensitivity, casein, dairy. So is that any part of some of the studies that you're looking at?
We haven't been looking at treatments for symptoms.
But maybe if there was a mother who was gluten sensitive or wheat sensitive and had inflammation.
We do have data on the mom's allergies and asthma, and actually I have a postdoc who has a paper that she sent me the last draft, and as soon as I get my next grant out the door, I'm going to read. We looked at the data, and there are some associations. I think there was it may have been the association we may have seen was with dairy, maternal dairy perhaps. But I have to go back and check and see what kind of fell out at the end after we did all the astute analyses. We also have a grant in that we submitted to try to follow up the kids in charge, the case group, to see what predicts children who improve over time and what predicts ones who don't, which ones seem more resilient. And I hope we get funded, because I think we've got so much information about their early life, and then we'll be collecting what treatments and services they got between early childhood and sort of the early adolescence period. So they're on the radar, those questions. Yeah.
Have there been any studies of older children or adults, retrospective studies, and have those been able to come to any kind of conclusion?
I don't think any of those have looked at issues related to environmental factors. There's a lot of debate as to whether there is an increase or not in autism, and people have been trying to do prevalence studies of adults. And some studies are suggesting that there's a lot of undiagnosed autism, or at least Asperger's, autism spectrum disorder. I don't think the final word is in on that, but there's some degree of underdiagnosis that may have gone on in past years. But that seems to be the main focus in the adult. And then the other aspect is, are adults with these conditions actually getting their needs, their service needs met? That's been the focus in adults. I haven't seen a study trying to then go back and look at those causes. Yeah.
Sort of a naive question conceptually over this whole idea. When we take the constancy of autism rates in all kinds of populations, and just, if you like, did a back calculation, what are the odds we wouldn't have a hot spot somewhere that shows us a specific environmental threat in all of those? Because I mean, one of the clear signals in autism is the relative uniformity of the frequency across population. Oh, here.
Not really. I mean, this Korean study came out with a 2.5% risk, and there's a paper coming out tomorrow that is embargoed that has a rate that's like about, way less than the U.S. rates. So probably about 4-fold less, if I recall.
So your statement is that you believe there are clear societal differences in the frequency.
I don't think well, either there's really major, and there may be that the diagnosis really differs a lot from one country to the next, or there may be environmental factors that are responsible for the geographic variation. I'm interested in environment not because I think there's huge geographic variation, but I think there actually is. But it's hard to tell. I mean, cultures are so different, and I think that even though we develop these reliable instruments, they're being developed within the context of certain cultures and certain behavioral norms and so forth. But the data are really all over the map in terms of prevalence rates, and the extent to which that's diagnostic differences as opposed to true differences, I don't think is known. I don't think we're there yet to be able to say that. But certainly the rates that are published are not consistent from location to location. Jim. Oh, Scott, actually, you've had your hand up forever. But.
Oh, yeah, just a quick question. I wanted to ask you about the really intriguing gene-environment interaction on the nutritional analysis. I want to know if there were other candidate genes you didn't show us.
Oh, we looked at, actually, I think it's a list of 8 or 10 that are in the one-carbon metabolism pathway.
Okay.
Some of them were stronger than others. The one that I showed you was the strongest, but there were a few others that were significant, and there were others that weren't significant, but they were kind of in the same direction.
Okay. So this one was statistically significant?
Oh, it was quite significant, and so was M.P.H.FR. Okay. And so was M.P.H.FR in the mom and M.P.H.FR in the child, and this was COMT in the child, but not in the mom. So.
And that was without adjustment for multiple comparison, presumably, usual.
Right. Right.
So if you would adjust for multiple comparison, or do you have a sense of, likelihood of type 1 error there?
I don't know. I can't remember. I mean, yeah. The thing is that when I'm looking at as a whole, this kind of pattern, it was always higher in the inefficient metabolizers that didn't take the supplements, but yeah. Jim.
I'm wondering, with your colleague Evan Eichler has been looking at the age of the fathers, and the notion of older fathers might be a risk factor for autism?
So there are data suggesting older fathers are a risk factor. In fact, there are data suggesting that, not in this one. In some of my talks, I actually have some age data after the thank you. So both older maternal age and older paternal age seem to be associated with autism. you really have to look at large studies, though, to be able to try to say, oh, is it the mom or is it the dad, you know? I think it's probably a little bit of both. We did probably the only analysis that actually can separate them out, and a lot of people say they have because they stuck both in the model and one of them came out or the other one came out, and it looks, you know, but the fact is that maternal and paternal age are highly, highly correlated, and to really separate them out, you've got to not model, but really stratify. And so what we showed was that in millions of births with tens of thousands of cases, we found that as mother's age increases, if you stratify in the youngest dads, the next youngest dads, you know, 10-year age groups, and we look at mom's age, is always an increase at every age of the father. If you reverse that and you take the strata of maternal age and look at the father's effect, you see a very strong effect if the mom is under 30. If she's over 30, you don't see the father's effect. There's a paper that has gotten so much press, it's always being cited for people who are really interested in paternal contributions to, genetic contributions to autism. And the study has a serious problem, which is that they didn't have maternal age on something like 2/3 of the people. So they actually had a much smaller, and it was a way smaller sample size than what I've talked about. It was, you know, maybe dozens of cases and thousands of population.
I have your next study is with offspring of mothers who already had an autistic child. And the other type of autism, I thought that Eichler and others were studying were sporadic autism, with no autism in the family, and you have a child with it. Have you looked at, and that was where I thought the age of the fathers might be.
Oh, in the sporadic.
Yeah, in the sporadic autism cases. And are you looking at that in any way, your charge study?
Yeah, so this is really, we're having problems with this because defining your sporadic cases is a little difficult. So you kind of have to take out anybody if it's an only child, because you don't really know. And then you look at the other members of the family, and we didn't have the money to actually evaluate all the other children in the family. So again, we're relying then on self-report of who else in the family is affected. And if the younger child is younger than the child who's in our study, we don't yet know. So we've been trying to organize going back a few years later to call all of these families and figure out, is this a simplex case or is this a multiplex? You know, is it sporadic or is it familial? And that's a big job. And it's, you know, we're still enrolling. So the most recently enrolled, you know, we don't yet have that variable to be able to put. But I think it's a very important question. Are there different factors playing a role if you've got, let's say, a genetically loaded family as opposed to one that's not? And, you know, I don't think we know yet.
I know why you had to look at mercury, but did you look at other metals that may have possible mechanisms of affecting brain function?
Well, we started out doing that, but it was a money thing, so we were focusing on the mercury. We also had some data on lead and did not see an association with autism. But again, this was measuring at the time point where after the diagnosis, so those measurements are post-diagnosis, you know, the life, the excretion is, you know, of both lead and mercury is on the order of, you know, 45 days for lead and 70 to 90 days for mercury, so half-life.
One last
Another
question.
question, yeah. Go ahead.
Early on you mentioned that there were some MRI studies. Are there any anatomical differences that you found?
So I haven't been heavily involved in that, but there are volumetric differences that have been seen, and I'm not the brain scientist to know, you know, which regions it was. There is a pattern of more rapid brain growth in the very early years of life, so that the brain grows more rapidly and then it kind of plateaus, whereas relative to typical development where it grows a little bit more slowly over time. at the, by the final growth, usually they're fairly similar. But on average, there does seem to be a more rapid growth, at least for a subset of children with autism. And I can't tell you what regions that's in, but that has been an area of great interest in fact.
Okay, on behalf of our students and faculty, we thank you for
Thank you.
presentation.
Some more.
Okay.
Thank you so much.